So, very welcome to our last speaker today in the afternoon session, Brent Doiron. And his talk is about how biological heterogeneity enhances cortical code.
All right, thanks so much. Thanks for the invitation to speak to you guys. This is my first Neuromorphic Chips conference. It's exciting. I'm a theoretical neuroscientist by training. We've already seen a lot of neuroscience today, so this has been a very interesting meeting for me. So I'm going to talk a little bit about how the brain actually does some computing here. We've known for a long time, really since sort of Hubel and Wiesel and before, that individual neurons, this is data from cat visual cortex, and they're looking at a particular neuron here, and this particular neuron likes drifting bars that have a specific orientation. And this is generally true for all neurons there. They all like a specific orientation. They fire a lot, and they fire less for other orientations that they're not tuned to. Tuning exists not only in sort of sensory systems, but in deeper areas of the brain. This is work from Patricia Goldman-Rakish's lab, where they're looking at tuning for a working memory task. It's still a visual input here, but the animal has to store in memory, short-term memory, where that input was, and this particular neuron here tends to contribute to storing sort of inputs to the upper left here. Different neurons contribute to different input tunings here. And this is work from my collaborator, Dave Friedman, whose data you'll see a little later. Neurons can be tuned to abstract data sets here, where they're, again, giving visual input here, but the monkey is forced to lump 3 orientations of motion together, and then another 3 together, and then we'll see that neurons in deeper areas of the cortex are tuned to the abstract category and not the actual motion of the bar itself here. So there's only two responses here. This particular neuron is coding for category 2 here. And sort of finally, in the hippocampal system, we've known for a long time, these both got Nobel Prizes here, where neurons in the hippocampus code for specific location along a track here. So neurons are tuned to the animal's location here. Okay, so we have this picture where there's a bunch of neurons here, and we have a complicated external sensory world that we live in. And due to sort of complex selectivity here, we'll get, for any given input, we'll get a distribution of firing rates from a group of neurons here. Some neurons will fire a lot. Most neurons won't fire very much. If you change the input the external world gives, then who fires a lot and who fires a little is going to change. And that's sort of the way that the nervous system does representation, sort of a sparse-like code here. If we wanted to sort of measure that, we could imagine taking, for a given condition, all the firing rates of the neurons, putting them along the diagonal of a matrix here, and then computing what some people call the participation ratio here. It's just looking at the mean firing rates squared, the mean over the population, divided by the second moment of the firing rates here. That's a number that's going to go between 0 and 1. If all the neurons fire exactly the same, 10 hertz, 10 hertz, 10 hertz, then it's close to 1. And then if neurons have a broad distribution of firing rates, like we tend to see in the brain, this measure will be less than 1 here. So if we want to come back to this picture, we're going to sort of measure the complexity of sort of a neuronal response by just computing this D of R here, this dimensionality measure of firing rates here. Okay, so this is, now we're going to imagine we're giving a stimulus here. This stimulus gives a response in some neuronal space here. So each axis is the firing rate or the activity of a neuron here. We've known for a long time that trial average responses are good to compute, but really if you have a population of experiments or a bunch of experiments, the response of the population is quite variable from trial to trial to trial. So there's this cloud of responses here. The inputs are exactly the same, and yet you've got serious fluctuations from a trial point of view. And this is something that I've seen in many, many talks here, this idea that the nervous system is unreliable, and there's a serious amount of stochasticity in the system, and we've thought about this for a long time. If we want to sort of measure this, we could imagine computing a covariance matrix of these responses here. When you have a matrix, you're going to get a bunch of eigenvalues here, and we can use a similar measure to talk about the dimensionality of this cloud here. This is the participation ratio. Again, instead of rates, we're going to use the eigenvalues. So we have the average eigenvalue squared divided by the second moment of the eigenvalue. Looks like this. And then if all the eigenvalues are roughly the same here, then you get this sort of isotropic cloud here. On the other hand, if one of the eigenmodes connected to one of the eigenvectors is large here, then that's going to lower the effective dimension of the system here. So this is a measure of looking at the dimensionality of the variability in the system. So if we add to our picture here, the way I want to think in this talk, there's lots of ways to do this. I want to think of external inputs driving trial average responses, which give us a neural code, and the wiring within a network here as being the primary source of how you get trial-to-trial fluctuations in the system. There's a long history in circuits neuroscience of thinking about the recurrent wiring in the system producing trial-to-trial correlations here. All right, so the goal of today's talk is to really link these two measures here. The distribution of average responses and the trial-to-trial fluctuations we see from a neural population here. And if we do that, we're really just taking what we use to code responses, the average response, and relating it to its noise here, and that's the first step of getting sort of a mechanistic sense of a neural code. Every code can be written down as a signal-to-noise ratio. This is, in some sense, the signal in the system, and then this is the noise in the system, and we want to have a way of relating those. Okay. So as an outline for my talk, I'm going to do what I just claimed to say I'm going to do. I'm going to relate these two things. We're going to test a prediction that comes about from this in a vast array of data across a bunch of different animal species, and then we're going to look at implications of this for population coding in the nervous system. So I'm going to do this in a really simple way here. We can do this, we can take spiking neural networks if we want, but for this talk, it's easy to do this in terms of a rate equation framework, and I'm going to take linear rate equations. So R, this is some vector of firing rates. They evolve linearly. There's going to be some input to the system, which will be stochastic, and then this is a recurrent weight matrix, and then I'm linearizing the system around some operating point. That's what L is. It's the linear gain of the networker. This may change with the state of the system. And once you have that, it's easy to get an expression for what the covariance matrix is. You have some input covariance matrix, C0 here, and eventually I'm going to take that just to be the firing rates of the system, assuming then the neurons are Poisson, so that their variance is equal to their mean here. And then this this is a diagonal matrix. This input matrix gets pushed around through the recurrent weights in the system to produce a non-diagonal, so a full rank C here at the end. Okay, so we have three random matrices here. We have L, W, and C0. And the question is how does the structure of the eigenspectrum of C depend on what we assume for the generating properties of these three matrices? And this ends up being a reasonably hard problem here. So let's break it down here. Let's take the simplest possible case. No coupling, and all the inputs are symmetric. So there's no wires, and every neuron gets the exact same input. In that case, it doesn't matter where you put, this is sort of a schematic for two neurons here. It doesn't matter where you put the neurons' firing rates. You're going to get this isotropic circular cloud of responses here, and the dimensionality of the covariance matrix is going to be maximal, sort of 1 here. This is the simplest possible case. If we now let the coupling here be on average 0, but the synapses are going to have variability. So this is heterogeneity in the coupling here, and G is this heterogeneity parameter. So the larger G is, the more random the synaptic connections are. What that will tend to do is, given a specific realization of this matrix, it'll take these circles and turn them into ellipses. It'll stretch it along a certain direction and compress it along another direction. And that ends up being, of course, a dimensionality reduction here. And actually, Moritz, who's somewhere here, and Eric Shea Brown and others, and Chaim Sompolinsky have shown that you can use replica method techniques to effectively get that you have this little simple equation here, that the participation ratio, the dimensionality, basically goes down with G according to this equation here. So it's a nice calculation here, and we sort of see that just heterogeneity in a system, unstructured wiring, just as a dimensionality reduction in terms of the variability here. Okay. Well, now let's go the other way. So this was still assuming symmetric inputs and symmetric gains here. Let's take the coupling away, but let's now let the input be a random vector here, so this firing rate matrix. In this particular scenario here, we don't have coupling, but if we move away from the diagonal, so if neuron 2 fires more than neuron 1, then the variability along the neuron 2 direction will be larger than the variability for neuron 1. That's the Poisson property. Same thing here if the other way happens. So heterogeneity in the firing rate responses of the system will also trivially drop the dimensionality of the system. But what I want to do here is, of course, I want to link these two sources of heterogeneity. Heterogeneity in the wiring and heterogeneity in the inputs here. And I'm going to make an assumption that the gain is basically proportional to the square root of the rates. That's effectively saying that the neuron's transfer function is a quadratic here. So neurons, but they're driven by larger and larger inputs. They tend to fire more and more and more like a square here. Okay? So how do we compute this quantity here of interest here? I want to tell you why it's hard, and then I'll not show you how I did it. So if we have two scalar random, so the problem here is there's two random matrices. There's the random matrix of weights and the random matrix of rates here, right? If I had two scalar random variables, and I said they were independent, and I was interested in computing a third quantity, C, and that depended on A and B like A, B, A, then if I want to get moments of C, then I just take the expectation of ABA, I commute, and then since A and B are independent, I just get this product of expectations that comes out. Easy. Right? If A and B are n by n matrices, where the entries are still independent of one another, and I play the same game, then it gets very difficult. So here, phi is some expectation operator for us today. Phi is just going to be looking at the trace of C or powers of the trace of C to get the eigenvalues. Anyway, it's just some expectation operator. We can't play the game we had up here because matrices don't commute. You can't just do this step here, bang to bang. And then as a result, it's difficult to get moments of the thing we're interested in terms of moments of the matrices we know. Right? Fortunately, smarter people than me have figured some of these problems out here. So there's an area of probability theory called free probability theory that says as long that A and B are freely independent, they define a new notion of independence that only works in the large n limit. Like a lot of machine learning, we sort of get a lot for free when we have large dimensional data sets here. So how do we compute this? Oh, sorry, I went forward again there. All right. There. So for n goes to infinity, in this particular case, the little example I had here, it actually does factorize into what you would expect in the large n limit given some conditions are satisfied. That makes it seem like basically you can do calculus just like you would normally here. That's generally not the case. Only in this little silly example does it work. If I just added another b at the end of my calculation, you can get an expression for this in terms of the moments of A and B, but it's not the one you would expect just from sort of classic probability theory here. So you actually have to do the math. We're not going to do it on a simple case like that. I'm now going to ask, okay, R is random, W is random, L is related to R, but I've still got two random matrices. So how do I compute the expected components of C in terms of what I know about the matrices? Bunch of math. And we get this cute formula at the end here. So the effective dimension of the covariance matrix looks like it's effectively the product of these two effects. We've got the 1 minus g squared we have to normalize because now we have firing rates. We have a gain in the system that's not 1, so we have firing rates. And we also have that the heterogeneity in firing rates also sort of decreases this dimensionality. So it looks like it's a product. You can only calculate this for this case if it's square root r. We've done numerics to show that that's not critical for the general form of this, but if you want that formula, you really have to have the square root here. Okay? So this gives us an opportunity to test our theory. So here we have what Eric and Moritz and company have looked at, and then here we have our extension where we've sort of broken the symmetry of the inputs and the gains here, allowing the inputs to be random here. If we want to test this in data, it's difficult. You can do it, it's difficult, mainly because looking at the covariance matrix and the distribution of synapses in a given data set is almost impossible. Right? It's you got to really look at a bunch of wiring. You can do this with connectomics. You can do it maybe in C. elegans. and a couple other components here, it's difficult to do that. You can make statements about looking at sort of path expansions in G here, but it's difficult to do that. Here, measuring the covariance matrix and the firing rates from the same data set is always trivial. If I can measure the covariance, I had to measure the rate. That had to happen, right? The covariance is just the second moment of the firing rates here. So this gives us an opportunity to sort of test this, namely that the dimensionality of variability is linearly related to how diverse the trial average response is. Okay, so we're going to see a lot of plots that look like this for the remainder of the talk. This axis is this firing rate uniformity. If neurons all fire at the same rate, we live up here. If neurons all fire with a diverse response, we live down here. This is the dimension axis, very low dimensional activity versus very high dimensional activity up here. If we sort of take this formula and just test it doing simulations of matrices here, unsurprisingly, we get this nice linear relationship between SIMS and theory. And how did we vary this axis here? So the way you should think about this is if I take two different points here, what I'm doing, or what my student John's really doing, is he's choosing a log normal distribution for the firing rates, generating a firing rate matrix, and for the same weight matrix, he's basically running this math here, and then getting a covariance matrix, and then looking at the eigenspectrum. We can actually get the full density of the eigenspectrum here, but we're really only interested in the first and second moment of it. And if we change the distribution of firing rates, so this one's broader here, then we change the covariance matrix and we change the eigenvalue distribution. So we should imagine the distribution of firing rates as being a knob that we're using to change dimension here. Okay? So let's test this theory in data here. So this is where I pair up with my collaborator, Dave Friedman. He has a non-human primate lab at the University of Chicago, and he's trained his animals to do these abstraction tasks. The details of the task aren't so important, but the fact that the task has a lot of different configurations, and each configuration will give us a distribution of trials, which we can then use to fit a distribution of firing rates for the 50 or so odd neurons he's recording, and a covariance matrix. This is what it would be for STIM1. We can have STIM2, STIM2 configuration 1, and so forth. So we can get lots of different conditions here so as to probe our system. So this is the response of the population to one of the conditions here. This is the distribution of firing rates. This is the covariance matrix. It lives here in my little plot. Different configurations, so this is now a different stimulus input to this particular group of neurons we're monitoring. The distribution of firing rates changes, the covariance matrix change, and so forth, and you live here. This is all the responses for all the different conditions that we have here. So we can see it lives on this linear relationship like we expect here. So that's nice. The linear relationship isn't trivial in that if the coupling in the system didn't matter, then we would just all live along the diagonal here, right? If G is equal to 0, who cares? We're just now looking at that trivial case. We definitely have a non-unity slope. The slope is less than 1, so we tend to have that both coupling, recurrent coupling in the system, and the distribution of firing rates tend to combine to effectively lower dimensionality here. This is the data you just saw. This is data in FEF, the frontal eye fields of the monkey. This is in visual area V1. This is visual area V2. This is visual area V4. This is prefrontal cortex. So across the monkey brain, we tend to see this linear relationship between the distribution of firing rates and the dimensionality of noise correlations. This is it in mouse, so all over the mouse brain. This is data from the Allen Institute out in Seattle here. We tend to get this linear relationship recovered everywhere. Lastly, I was fortunate to work, be able to collaborate with my late colleague Sleeman Ben-Smaya's group at the University of Chicago. He has a human brain machine interface lab here where he's got 2 participants that have chronic Utah arrays implanted in area M1. These participants are trained to do they just do reaching tasks here, and we're able to record populations of activity in area M1. This is what we see. So this is participant 1. This is around 63 neurons. We get a really nice relationship here. This is the second participant. We have 80 neurons. We get an okay relationship here. So this scaling also works in humans. So monkey, mouse, and human brain areas all tend to show this same relationship. So then in sort of the last 10 minutes, I'll sort of ask, well, why would you care about this? What's the point of this? It's nice that it happens everywhere, but should it matter here? So let's think about population codes here. So here we have schematic of 3 neurons. You can imagine 2 stimuli coming in. Stimulus 1 drives this 3 neuron population to on average live here. Stimulus 2 drives them to on average live there. So if we want to put a constraint and say, okay, let's say that for any given stimulus, the firing rates are allowed to be variable, but the sum of the firing rates has to be a constant. So there's some sense of conservation here. That means the responses has to live somewhere on this triangle. On the other hand, if we say, well, what if the sum of the squares of the firing rates is equal to a constant? So well, now it's going to live somewhere on this quarter sphere here. If we ask both of them to be true, then we can look at the intersection, and the intersection is going to be this circle. This would be a n-2 hypersphere in larger system size. It's this circle, so if I'm asking for the mean response across the population and the second moment of the firing rates to be constant, then you have to live somewhere on the blue circle here, right? And asking these things to be constant is basically asking for the participation ratio or the diversity of firing rate responses to be equal to a constant. That's why I'm doing this here. So what does it mean for this to have a certain number here in a population coding sense? It means you sort of restrict where you live. You have to have your responses live on this circle, not anywhere they want in the space. So if you have a large DR, which means you're going to have a very homogeneous response, a lot of neurons the neurons tend to fire at the same firing rate, then this circle will be quite small. If they fire at the exact same firing rate, then it's only the center here. And then the more heterogeneous the response is, sort of the bigger the landscape over which you can tile responses for different stimuli. So here's again the response for stimulus 1, it might live here, and the response for stimulus 2, in this particular case, there's going to be a larger distance here, and you can sort of imagine from a capacity point of view, this is going to be superior. This is basically the idea of mixed selectivity or sparse codes in the nervous system, that you can have a large capacity here, whereas if you force all the neurons to have similar firing rates for every different stimulus, then it's going to be hard to code lots of different stimulus because they're going to overlap a lot. Now that was me playing with this axis, but we know that the uniformity of firing rates also controls the dimensionality of the covariance matrix here. So now I'm going to look at lots of trials here. So not only do they live closer to one another on this blue circle here, they're going to overlap more. The chance of their overlap is more because they're higher dimensional. Whereas here we're going to have sort of lower dimensional activity just by virtue of the fact that D of R is large. Small. So you have a broad distribution of responses here. So lots of work has gone in to show that if you have low dimensional noise, then it's going to be easier for a linear classifier. If you have the noise is unstructured and not related to the signal dimension, then it's going to be easier for you to put a linear separator between them. So a classifier is going to have an easier time classifying if the noise is low dimensional here. So we can take our framework here. So here's our 2 stimuli. We can sort of do very straightforward stuff and ask what the D prime is. So the D prime is some discriminability index here, and it's really asking for the centers of the clouds to be as far away as they can. That's this R2 minus R1, and you also want the variances to be as small as they can. So you're dividing here in some sense by the variance here. If we sort of assume log normal distributions of firing rates, we can basically calculate the following relationship for D prime just in terms of the distribution of firing rates here. This first term is really just that the means are separated. Remember, when D of R was smaller, the circle was bigger. That's better for coding here, and this is exactly what this is. So the smaller DR gets, D prime gets a boost here, and then this is the effect of DR having on the dimensionality of variability. It also gets a boost. So there's sort of a double whammy here, where you get an improvement of codes as the population of neurons gets more diverse. You have 2 reasons that codes get better: better capacity, and then the noise is easier to deal with from a decoding point of view. So we get this. So as we change the firing rate uniformity, we get this discriminability that gets better and better and better. Let's test this here. So in data from the Allen Institute, we were looking at around 150 neurons, and depending on, we had the firing rates of the neurons, and we were trying to decode what the stimulus was that the animal was driven by, and depending on the distribution of firing rates for that particular group of 150 neurons, as we were changing the stimuli that gave them a broader spread of responses allowed us to better discriminate between two pairs of stimulus here. So that's sort of similar to what we have here. That was one experiment. This works in general for most mice. That's just the slope of that line here. We want the slope to be negative. For most of our data sets, it is negative here, which is nice. But that's not really proof. That's us having access to 150 neurons and then playing a game where we're decoding those here. The animal didn't have any, it was good at decoding all different orientations. It wasn't specific to any particular pair. It was specific for our pair because we were really massively subsampling the network here. So what about, is there any behavioral evidence that the brain is actually using this? So let's go back to some of that monkey data. This is data from the frontal eye field here. So here we're plotting it here. I didn't tell you that half of the trials ends up just being the animal looking at sort of a blank screen here, looking at spontaneous activity. So versus this is the scenario where the animal's actually doing the task. So we can see a brain state change here. When we go from spontaneous or non-driven activity, we tend to have more homogeneous responses, higher dimensional variance, and then when the animal's asked to do a task, the brain shifts the system here, in part likely because it's changing the distribution of firing rates in the evoked state, and then that lowers the dimensionality of the system for free here. That's what we have, and this is where you want to code better. Remember, we want to have smaller D of R to do better codes. The animal shifts its network of neurons to do exactly that. This is data from the Allen Institute. This is data when the animal is looking at stimuli that it didn't care about, versus when the animal was looking at stimuli that was connected to a task. So if it got the task right, it would get juice here. And we saw the same basic effect that we have a better coding system here when the animal actually has to code using its visual system. And then this is data from the Allen Institute when an animal is looking at visual images, but it's just sitting still versus when the animal's running here. And it's known that the visual system of mice in particular, it operates in a more coding-rich framework. When the animal's running, it doesn't want to bump into things. So it's running, and then there's top-down processes that drive the visual system to sort of take advantage of these broad distributions of firing rate, lower dimensional variability here. Right? So this is basically saying that the brain seems to take advantage of the fact that it can shift its distributions of firing rates to be in a regime where you have low dimensional noise, and that's better for codes here. So in the sort of last 2 minutes here, this is anecdotal. So what the paper we're writing finishes where I just stopped here. This is the human data here. So generally, you normally can't release names, but I have permission to release this guy's name. His name's Scott. So I've met Scott. So he has, again, an implant in M1. So Scott has the opposite. So in his spontaneous state, his firing rate uniformity was lower, and in the dimensionality was also lower. So it agreed with our theory here generally, but it didn't agree with this spontaneous evoked. So when we looked at this data, I went and I talked to Scott, and I said, what was going on during the experiment? So, of course, it's this evoked task. Scott has been doing this for 2 years. Monkeys, when they do this right, get juice. Scott gets paid no matter what. So he doesn't have, he's not driven to get the task right or wrong here. Also, because I needed covariance matrices, I made them do a reduced number of conditions, but way more trials per condition, so I could get a sampling of the covariance matrix. So all of that sort of led to Scott being, let me see here, there, Scott being bored. Scott was bored this. And then I said, well, what are you doing in spontaneous? So remember, this is the motor cortex, so as long as he's not moving, it's spontaneous. Well, they let Scott watch Netflix during the spontaneous, and I asked, what were you watching? And he always watches these National Geographic shows where some lion's taking down a gazelle. Very exciting. It's possible that he was certainly more engaged during the spontaneous. Even though he's not moving, he's watching movement, and he's probably planning movement in his motor cortex. So here, this is sort of interesting here that, what is the nature of spontaneous activity in the brain? It's sort of in the eye of the beholder here a little bit. So this is sort of a fun ending here, that even though our theory is still right, generally here, we have to talk about what motivates an animal or a human in this case here. So in summary here, basically what I've shown you is this cute little formula that relates the dimensionality of noise to the distribution of trial average responses. And this has widespread support across a bunch of different brain regions. And by lowering the distribution, by making the distribution more broad, dimensionality gets lowered, and this seems to correlate with a lot of how the brain is changing its internal state. So this was all done with my fabulous grad student, John Tien, and here's my group, my funding, and I'll thank you guys for paying attention.
Thanks, Brent. Perfect timing. So, time for questions from the audience.
Sure, I can hear you, yeah.
So I was wondering, Brent, whether this is somehow related to Anna's talk earlier with the two time scales, and that's the first question. And the second question is exactly this, and she was talking about attention, that maybe the human was more attentive in this situation rather than avoiding the repetitive reach-out movement.
That's a good question. So I've collaborate with Marlene Cohen, who also looks at area V4. So we've analyzed Marlene's V4 data during attended and unattended regimes here. So I think sort of the spontaneous evoked is more about, that's about sometimes arousal states, and that changes the whole brain typically here, right? So where these fine controlled attention experiments, the animal's always engaged, it's always doing the task, it's just whether stimuli are in the receptive field of the neurons that they're recording, or where the attention is directed outside of the receptive fields. So when we've looked at this in Marlene's data, we haven't seen such a clear segregation here in Marlene's data. It seems like attention is much more subtle on how it changes firing rates compared to Scott doing something boring or watching Netflix. That's a whole brain state change, and that seems to drive this effect much better.
Hi, so I'm Matteo from the Institute of Neuroinformatics in Zurich. Thank you very much for the talk. I had just a question. You showed very nicely this linear relationship between the two matrices, and you were able to have the theory behind that, because you show like you had this free
probability.
Yeah, well, the freely independent, I don't remember exactly the wording, but this different way of defining the matrices to be independent.
Right.
But given that it's a very nice but very simple also linear relationship between these two metrics, do you think there is a more parsimonious way to get exactly the same results but without having to pass through all the math you showed?
So quick answer is no, so no. And I have a couple slides to sort of, I think talk about this. So I found this everywhere, right? I found this everywhere, and then you're saying if you find something everywhere, there should be an easy explanation. You shouldn't have to go to graduate level.
I was just wondering, yes.
Absolutely. That makes total sense. So this is the recordings from Dave's lab, FEF, LIP, and this is area SC, that's sort of lower here, subcortical. SC is not as great, but it still has a correlation coefficient of 0.43. So in each of these areas, we basically assumed that we had sort of an IID distribution of weights here. What I didn't tell you is Dave actually records all these areas simultaneously. He has 3 different probes in the brain simultaneously. And this comes from looking at the FEF block. This comes from looking at the LIP block. This comes from looking at the SC block here. Okay? But since I have the full matrix, I can ask, what does it look like for the full matrix? And it's horrible. It's horrible. It doesn't work. So when you try to play this game here, by just concatenating all of the neurons together in some grand network here, and then ask if this works, no. And I'm happy that it doesn't work, because this was already a suspicious assumption. We know a lot about the wiring of neurons within a brain area. We think this is probably a pretty poor representation of it, but at least in the context of this theory, it worked okay. But if it worked for the full system, then that would be saying that the wiring within an area is the same as the wiring between areas, and the cortex is just a soup, right? There's no structure. All the anatomy we've known about for hundreds of years or 100 years is not true here. So the fact that this fails tells me that this is not just a generic property of just neurons that we record. It has to be neurons that have similar connectivity rules, which probably means it's local to a brain area. Does that answer a little bit?
Yeah.
Thanks.
Thanks.
I know.
I can also speak loud.
Very loud.
Connected to that, maybe, like what I was looking at is time scales,
Uh-huh.
different brain areas, slower time. Like I don't know how this areas to like connect. Like to some extent, slow time scales, like say, are closer to the second or the phase transition because if I look at the scale, this is larger variability. This would mean that in your representation, the areas that are more slow time scale frontal would have maybe stronger dependence even, or at least like be shifted compared to the other one.
That might be lower dimensional, so we can kind of, maybe we can go, I don't know if I can go all the way back there, but on our cloud here, I can look if prefrontal cortex has lower dimensions than V1, let's say. So I don't know if I can go all the way back. It's not like it's frozen me out here, but we can look at it together. I don't know if that's the actual result, but you're saying slow dynamics might mean more global within that group, and then lower dimensional, so then those cloud of dots should just be translated to a lower value in prefrontal versus V1, or maybe there's some signature of slow time scales and low dimension don't have to go together, but maybe in recurrent networks they do. Yeah, it's good. We can look at it.
Any further questions? If this is not the case, thanks again to Brent Doiron.